generation points in both the organic and inorganic rates, so I can't show that. But for calcification rate, so the uptake for inorganic material, we again see an increase in the future ocean conditions, and again around 25%. So that fits quite well with the content we have seen. As the last data shown, it's the inorganic to organic carbon ratio, or the calcium carbonate to organic carbon ratio. We see a trend of an increase in future ocean conditions, which makes sense because we saw more calcium carbonate in the future ocean conditions, but no change in the organic carbon. So that fits also well together with that. And then just to sum up the results, because I showed quite a lot of them, so the nitrogen limitation in both treatments was at the same level as we have seen with the C/N ratio. Chlorophyll concentrations were higher in the elevated CO2 and temperature treatment. There was no change in the organic carbon content and production between the treatments, but we saw higher calcium carbonate content and production under future ocean conditions. The PRC to POC ratio seems to increase under elevated CO2 and temperature. And last, we didn't see any change in the response after 200 generations. So 200, 400, 700 generation points were all the same. There are more data to come, which I hope will probably help me to build a better picture of where the carbon did go in our chemostats, how the carbon cycling was in the chemostats. So we will still have to measure dissolved organic carbon and transparent exopolymer particles, which are both more or less the carbon that cells will release again after they took it up. Not using everything to build organic material, some of them is just kind of like released as organic compounds. And then also we will still measure cell size and look at the coccolith morphology, which means we look with an SEM at the coccolith or the cells from the outside and look if the liths are all shaped the right way. They are all built inside the cell, so if something in the cell during the calcification process goes wrong, you would see that these liths start to be malformed or incomplete, stuff like that. So I want to look at this too. And with that, kind of like trying to get an idea what all these data mean. So if we look again at the table with the 3 studies done already under CO2, elevated CO2 and temperature, and then look what changes we saw. So first, we also saw no change in the organic carbon content and production, but we saw an increase in inorganic carbon content and production. So same results for the organic part, but opposite results for the inorganic part. And then now with 4 studies all showing the same for the organic carbon, I wanted to compare that to the results we have seen so far with just CO2 treatments. And if you remember this small table with all the different responses, but one interesting part was that the majority of the studies done have seen an increase in photosynthesis in these studies. So we see kind of like different responses for just CO2 treatments and CO2 plus temperature treatments, which could be interesting to go more into details and see if there are synergistic effects of the two. Then in terms of the inorganic carbon, I compared our study to one short-term experiment done by Feng et al. These are the results they found, and I choose this one because they use the exact same strain we did, they use the exact same temperatures we did, and they use quite close CO2 levels to ours. And then if I compare it to our results. So we see the same for photosynthesis, opposite effect in calcification. So there are a couple of possible explanations for this. One could be the number of generations. Feng et al. Was a short time, so they only kind of let them grow for around 10 generations in the experiment. Our results were seen after 200 generations. There's a possibility that over time, the cells started to adapt to the new environment and maybe flipped the response from decreasing to increasing. But there were other differences between the experiments. One big difference is that our experiment was nitrate limited. Theirs wasn't, because they were just growing them in bottles. So there's also the possibility that we have some synergistic effects with nutrient limitation, CO2, and or temperature. So there's still quite a lot of possibilities what could be responsible for it. And with that, I'm at my second last slide. So open question, things I think we still need to know about ocean acidification and coccolithophores. One is still we are not exactly sure why some strains, why we see enhanced calcification in some strains and decreased calcification in others. So we're quite sure right now it's a strain-specific response. But what makes the strains differently responding? And we have seen that adaptation is possible, but the question is again, would the different strains react also differently or adapt differently? One possibility could be that if they come from different regions in the ocean, they might also have a different potential to adapt to the new environment. And then, as I mentioned with the slide before, there's still a high possibility that all kind of different environmental parameters might have a synergistical effect. So future ocean will change a lot. It's not just CO2 and temperature, it's also light, it's nutrient levels, different nutrient species ratios to each other. We don't know how much they all have a synergistical effect. It might even change the whole picture. Again, what we think nowadays we know about their behavior in the future. And then the last part is most studies are done with Emiliania Huxleyi, which is just one species of coccolithophores. There are other species that are also really important in the environment. There are also big bloomers in some parts of the ocean. And we know more or less nothing about them. There are barely any studies done on other species. And with that, I just want to acknowledge the PIs of this project and a couple of people who helped with the project and my funding source. And thank you for listening.
Thank you very much. Can you describe just a little bit how the length of time that was involved there? Was the dilution rate set for approximately a doubling a day or something like that?
A little bit more. We had a growth rate of 1.1, which is around 1.5 doublings a day.
So you watched it basically for a year and a half. Is that how it worked?
Yeah, it was around 16 months, as I said. It's
the same culture. You just turned it on.
Yep.
In a big,
and it
flurry of activity, and then
we just keep it running. Yep.
And then you just watched it for.
Yep.
So at the end, if you were to have taken, could you tell the difference microscopically between the look of a coccolith that has more calcium carbonate on it than one that doesn't, which is I think what you were saying you found, is that correct?
Yeah, so under future ocean conditions, I found more calcium carbonate per cell. And yeah, I started looking at, with an electron microscope at the lith. I don't see any difference in the lith. What I haven't checked so far is there's a possibility that they had more lith per cell in future ocean conditions. So then they would have more calcium carbonate per cell. it's a little bit hard to check that with an SEM. I still have to work that a little bit out. So there's a possibility that they had more lithium per
But
cell.
of all the things you would have predicted, I mean, did it surprise you that you wound up with the result that is opposite of everybody's textbook anticipated result, that you increase the CO2 and therefore you decrease the precipitation rate and you should be losing your calcium carbonate, and you gained it?
I was surprised because, like I showed, there was a short-term study and they saw decreasing calcification. Even if kind of like I thought maybe they might start to adapt to the new conditions, I would have thought maybe I see less of a decrease or no change. I didn't expect to see an increase. And like I showed with a table of studies done with just CO2, some of them show an increase. But yeah, in the community, they're still kind of like heavily discussed. A lot of people don't believe that they might increase. What you mentioned with kind of like the precipitation of calcium carbonate should be harder under increasing CO2. One thing is with coccolithophores, they build the calcium carbonate inside the cell in a vacuole, so they can control the environment. The question for us is more kind of how much can they control the environment. It's not like other species that actually more or less just calcify on the outside of them, so they're definitely way more but changes in the seawater have a bigger effect, should have a bigger effect on them than inside the cell, where the cell can more control the environment.
Yes, and I wish I knew more, but I don't. But is there another organism that shows a good example of this? If you watch it for a year, it does what you're not supposed to. It does what it's not supposed to do. Like, is there a coral observation in which theory says it should lose calcium carbonate and instead it gains it?
I haven't heard any studies showing for corals.
Is this the one and only?
So, as far as I know, the one and only long-term so far, yes. Like I said, there are some short-term experiments with some strains who have shown also an increase. But yeah, it's heavily discussed, and that's why I'm kind of like going through all the different possibilities I can see in my data, because I know that a lot of people will not be happy with the result because it goes against what most people have seen.
It's fantastic because persistence maybe pays off. You stare at it for a year and a half.
Now, maybe we all have to stay at the ocean.
Yeah. Has anyone looked at genetic diversity of Emiliania Huxleyi around the world? Do we know how many strains there are in different areas? I know that you were saying geographically the adaptations could be different, but do we know how diverse they are, how many strains there are?
We have no idea how many strains there are, because in reality there's a possibility that every cell you take out is differently. So there's an endless possibility. Studies have looked at genetic diversity, and they have found quite a big diversity. Actually, I know that a lab looked at Emiliania Huxleyi and at a really close relative of them, Schiefer-Capsa oceanica, and they found some strains were closer related to the other species than to other strains of Emiliania Huxleyi. So there's quite a big diversity, which makes it even harder, to kind of like, then how should we expect to see the same results all the time?
Because the liths are accreted within a vacuole, the organism has a fair amount of biochemical control over the process. Do you know that the calcium carbonate is being formed from the carbonate in solution, or is it actually being formed from the bicarbonate or the CO2 itself?
No one knows what exactly happened in the vacuole. What we know, and people have shown with isotope work mostly, that the cell takes up bicarbonate, which then goes in the calcium carbonate. But if it's then converted first in the vacuole to carbonate, or no one exactly knows. So all we can say is, in general, what they use from the water is bicarbonate. That's all we know.
predicated upon a reduced carbonate ion concentration. But they don't care about that if they're taking it from bicarbonate. So I think that your results are very consistent with the real chemistry of the seawater.
It's one of the things when I started during my PhD, I worked a little bit on ocean acidification and a lot on coccolithophores already. And yeah, looking at all these papers, it's always started with, oh yeah, with ocean acidification, we will have less carbonate, and with that, calcification for these will be more hard. And I was like, but this paper tells me they use bicarbonate. So I was always surprised about that too. People use it as a good introduction to explain why they're looking at these organisms. But yeah, like I showed, it's kind of like it's barely any change in bicarbonate with ocean acidification. So why should they really care? So I think the only thing here in terms of calcification is the increase in protons, because it might mess up all the kind of like different pathways and enzymes in the cells.
This lab has a long history with iron limitation and its climatological effects. Has anybody, going back to that last slide, has anybody looked at using iron as a limiting nutrient?
There's one study they looked, I think, just iron limitation and calcification. I can't tell you the results anymore. I would have to look them up again, but yeah, that's one study. And I looked also actually for different metals, if I could find anything else, and iron was the only one study about iron that it is.
I was curious, for the other studies that have been done with Emiliania Huxleyi, did they all use steady state systems like yourselves as far as how they cultured their algae, or did they do them in flasks where the nutrient levels could drop based on growth or whatever?
So most of the short-term experiments are done in flask, but it's diluted batch, so they don't let them grow to high cell concentration. They harvest them when they're still quite low and are sure first, all the nutrients are still repleting. And also they haven't changed the CO2 system in the water too much, because they take up CO2 and they calcify. If you let them grow to quite high cell concentrations in a bottle, they start to change the whole carbonate system by themselves already.
Okay, so they do it before they reach senescence?
Yeah.
Okay, and the lighting regimes are the same across all the studies, or does that change also?
Mostly, that's kind of like one of the things, kind of like, I think we should be able to see a pattern, kind of like why we see the different responses other than just strains. But because every lab does it a little bit differently, it's really hard to compare them all to each other.
Wasn't there just something inside of you that wanted to like sneak a peek when you started the high CO2 going? Didn't you want to just like after 10 days go, now what happened to them? Instead of waiting for 200 days?
Yes, we plan to do that, to have kind of like also kind of a short-term experiment. Problem with the chemostat is it kind of like takes them a while to get stable. And I was kind of like, so I'm waiting till they're stable, and then I'm waiting a couple of days and take samples. But you're also, they're stable for 5 days, so I should start counting. And then one of them dropped again, so I was waiting again. And then at one point, I was way over the time point. I was, okay, now I'm just waiting for 200 generations.
So you had mixtures of tanks. Were you metering pure CO2 with air, or did you buy from a manufacturer pre-mixed CO2 percentiles?
We mixed ourselves. We have a CO2 system where we mix air with pure CO2. Yeah.
I see. Okay. And that was from a cylinder, basically. So
the CO2 is from a cylinder, yeah.
And did it last all 300 days?
Oh, yeah.
Oh.
We still had tons left. I mean, you don't need a lot of CO2 to increase from 400 to 800 ppm. Yeah. That's kind of like a tiny little percentage of the air, actually. So it lasts quite long.
You chose CO2, though, to look, and you think it makes all the sense in the world when you use CO2 to look at the CO2 effect, which is sometimes inferred as a pH effect. Other studies have just said, well, I don't want to deal with the CO2. Why don't we just alter the pH and then measure all the carbon species and see what the effect is? Would that, if you had created, do you think, I same, I tried to memorize it, I think when you went high CO2, your pH went down to 7.6.
Yeah, 7.7.
If instead you had titrated HCl in and achieved 7.6,
I personally think I wouldn't have seen a difference because what I think mostly affects them are the protons, and changing the pH is the same result. There are actually a couple of studies are done because people started doing CO2 experiments with just adding an acid because it's easy, it's cheap. But then people started to discuss, but you're not changing the carbonate system in the same way when you add CO2, which what happens in the ocean. And it lasted quite a while that people did both, and then we're kind of more or less fighting each other. Kind of like what you do, you can't compare to my results, and so on. And then actually last year, finally a study came out where they tested both. And they found the same responses they tested 2 strains, and they saw the exact same responses with the 2 different methods of changing the carbonate system. One more kind of like, I think, result showing that most likely it's not the different carbon species that are important for them, it's the protons in the water.
Okay, I think we can